unmuted. I'm going to leave it that way, okay?
Good afternoon. I'm Mike Duffy, president of the Cape Cod Tufts Club, and I would like to welcome 113 participants to today's program. That is a phenomenal number. In addition to members of the Tufts community from around the country, we have many members from the intercollegiate alumni clubs of Cape Cod, including alumni from local schools such as MIT, Harvard, Cornell, and Boston College. Through your generosity when registering for this event, we have been able to raise $760 for the Cape Cod Tufts Club Scholarship Fund. This fund allows us to award 3 $2,500 scholarships per year to deserving, currently enrolled Tufts students from the Cape, Islands, and South Coast areas. A few housekeeping items before we start the program. This meeting will be recorded and uploaded to our Tufts Alumni YouTube channel. You will notice your audio and video are disabled. Once we begin, you will see the speakers on your screen. You'll be able to hear them, but they will not be able to hear or see you. You may need to adjust your volume. If you would like to ask a question during the presentation, you can do so by clicking the Q&A button at the bottom of the screen. This will open a window where you can type your questions. After Dr. Hanlon's presentation, Amanda will read the questions for Dr. Hanlon to answer. We will do our best to get to as many questions as possible, but we may not be able to answer everyone's question. Now I'm going to pass the video off to Dr. Jack Whitehead, a member of our board, who will introduce today's speaker.
Thank you, Mike. My name is Jack Whitehead. I studied mechanical engineering at Tufts University, and I fell in love with fluid dynamics, and I continue to be active as a senior scientist emeritus at Woods Hole Oceanographic Institution. My wife, Lynn, and I have lived and raised a family on Cape Cod and Falmouth near Woods Hole Village since 1971. Our speaker, Dr. Roger Hanlon, has lived here too for about 25 years, about a half a mile away from us. Dr. Hanlon came from the Cincinnati, Ohio area and attended Florida State University, where he studied biology and played baseball and football. Then after the Army and travel, he earned a PhD at the excellent Rosenstiel School of Marine and Atmospheric Sciences at the University of Miami. After work at Cambridge University in England and at the University of Texas and some sabbaticals, he joined the Marine Biological Laboratories, we call it MBL, which is part of the University of Chicago, in 1955. He runs a highly active laboratory as a senior scientist and is also associated with Brown University and the Rhode Island School of Design. He has co-written 2 books, bunches of book chapters, well over 200 scientific papers, and is cited more than 15,000 times, has had numerous students and worked with innumerable collaborators. His work began out of curiosity when he looked at an octopus on a coral reef in Panama in 1968. As great scientists do, he has continued to follow his curiosity, and he's made great contributions to fundamental biology with highly practical benefits to boot. He has overlap with art, advertising, cosmetics, camouflage, etc., as you will learn. Roger?
Well, thank you, Jack, and thank you, Mike, for the invitation. It's a real pleasure to be here and to talk to all of you and hopefully entertain you on this beautiful Saturday. So I will maybe explain the title a little bit. The Octopus is Tech, and we're looking at the technological side of, let's say, the outcomes of some of the basic research that we do. And I'm basically one who does sort of pure science, but I get involved in applied science just a little bit. But I'll move right into it because I think the underwater world is going to tell the story here. So I'll take you diving right away And here you see a rock underwater and a fish swimming around. It's a very boring scene, nothing happening till now. And you see that there was an octopus on that rock the whole time. And now it inks my face and jets away. If we look at that slow motion reverse, you see a ring will develop around the eye. Here it comes right there. That's 5 million chromatophore organs, pigmented organs innervated directly from the brain. Another 25 million create the pattern. Look where my cursor is on the right side of the body and watch the skin morph from smooth to three-dimensional, a very important aspect for good camouflage. So the before and after is really dramatic, and they do this very quickly, as you noticed. Now, this animal can go to the top right where you have a more complex coral reef, or to a fully developed coral reef and move around anywhere on that reef, and in a fraction of a second can look at the visible background and can make decisions on which type of camouflage to use and to implement it. So it's this visual sensory motor system that I've been very interested in for all these years. And there's actually an octopus in the middle of that image right there, if you can see my cursor. So the camo is very good. Well, this is the subject I talk about, rapid adaptive coloration, and it's used for camouflage, as you've seen, also for communication. And down at the bottom, we're really slant to talk a little bit towards how some of these aspects can inform technology. Wouldn't we all love to be able to change color and pattern, either in our clothes or our car or our complexion or whatever? Well, here's a cuttlefish I filmed at night in Australia, and you notice the patterning change going on. They're little swirls of color. There are horizontal thin stripes and horizontal white stripes. Now we can go kind of yellow and put a bar across the middle of the body. And now it decides to plop on the bottom next to a coral and go camouflage. And we have two squids here on a coral reef in the Caribbean. There's a male on the left, female on the right. And the males fight all day long among other males to get a chance to pair with and mate a female. And here's how the fight is conducted. You see these two males fighting, and they put patterns for communication on their dorsal surface and also on their ventral surface to give different signals of communication. And the female is out of the picture at the moment. We're going to see who wins the very gentlemanly fight right here. And there's a little nip at the end coming right now. And now the winner swims off and stays with the female. And when he does so, he guards her. And we have the male on the left showing a brown courtship pattern to her and a white aggressive pattern on the other side for approaching males. Watch what happens when she changes places. Watch the guy at the top. And you see that he shifted his aggression pattern to the other side of his body. So these animals have very fast skin control, but they can do it even in the context of sexual selection. So this is what, this takes a big brain, a lot of cognition. So this is not a snail, even though these animals are mollusks in the same phylum. Here's the Broadclub cuttlefish showing a dramatic fast pattern, passing cloud pattern. They do that towards a potential prey like a crab, and we think that it mesmerizes the crab just long enough for the cuttlefish then to eject these two fast tentacles and seize it and have lunch. And I just want to make a brief say about the tools and methods that I use in my research. I'm basically a diving biologist. We do a lot of lab experiments. In the top layer here, you see lots of cameras, spectrometers, and so forth. I do use instrumentation in this middle row, like AUVs, autonomous underwater vehicles on the left, or ROVs, remotely operated vehicles in the middle, spectrometers. And we culture animals in the laboratory down here and do a lot of laboratory experiments, which I will show you briefly, and a lot of microscopy. But I want to point out at the bottom right here, this is one of the most important tools of all, and that is a good human observation and thinking about what's going on. So just a word about the brains of these animals. They have large brains. On the left is this very elegant dissection of an octopus brain, and this large central mass right here is illustrated on the right. There are 34, 36 lobes of the brain and millions of cells. So this is a complex and sophisticated brain by any measure, vertebrate or invertebrate. Here's a diagram that shows the peculiarity of the nervous system of the cephalopods. This blue spot in the middle is where the central brain is, right between the eyes, and you just saw a drawing of that. There are about 80 million neurons depending on how you count those. But what's incredible is in the rest of the body, the peripheral nervous system, there are 300 million neurons. So this is a nervous animal, so to speak. It's got neurons everywhere. And most of them are given over to controlling the 8 arms and these millions of color chromatophores in the skin and so forth. Interestingly, that even though you have a central brain, at the base of each arm, there's a little, we could call it a small satellite brain. So some people could count and say, well, they have 8 small satellite brains and 1 central brain. And the big question that we and others are looking at now is which controls which, and we don't know the answer to that yet. But the point is the brain is very sophisticated. Well, that brings us to a quick evolutionary context picture that I want to show you is that we've got mammals on the right, that's our side of the evolutionary tree, and on the other side, you've got the cephalopods back here. The common denominator for those 2 groups was a long time ago down at the bottom here. And the curiosity question is, it seems as though that the cephalopods have produced complex behaviors in a different route than we mammals have. And the big question is whether or not the cortical structure of the cephalopod brain is fundamentally different. Now, if it is, that tells us that there's one more route to evolving intelligence on the planet. We don't know if that's the case yet, but it's a big question that evolutionary biologists and neurobiologists and animal behavior folks like me are looking at. And I think if it turns out to be the case, then the artificial intelligence community would have one more model other than the human model for complexity and intelligence. But I'm dreaming a little there, but a lot of us are looking for those blue sky questions. Now, 2 discoveries recently about the cephalopods that are at play here. One is that their genomes have been done just recently, and the genomes are absolutely enormous. That is a big unexpected feature. And the genome is loaded with things for the nervous system. That's not unexpected. But what's really curious about cephalopods versus any other animal group is that instead of just taking their DNA and making a transcript of RNA and making a protein, which is how all life works, the cephalopods take that RNA and they edit the DNA at an incredible rate, 500 times more than humans do. It's really an amazing difference. We don't know what it's all about, but it sets them apart in terms of how they deal with genes. And most of those are for the nervous system, it's thought. So again, might explain a little bit about why they aren't a snail any longer. They're very different. Now, rapid adaptive camouflage requires keen vision so that the animal can look around it and see what the microhabitat looks like, so it decides on the right camouflage pattern. And then, of course, what I'll glibly call magical skin, and you can decide if it's magical after you see a few images. But to experiment how they decide on camouflage, we go in the laboratory. This is a cuttlefish in the middle right here, and then we give it some kind of visual background environment, which we measure quantitatively. And then the answer comes out in the skin in some kind of body pattern. And so this is a look at the brain. It's a visual look at the brain because the body is showing you what the brain decided in. This is a great advantage for looking at the brain without dissecting it apart. We put the animal on uniform background here, and it's a uniform pattern. If we give it a non-uniform pattern, a small checkerboard pattern, it's changed its appearance. And on a large checkerboard pattern, which has solely increased the size of the checker and the contrast, the animal is morphed from a uniform camouflage pattern to a mottled pattern to a disruptive pattern. And so what this means is we can probe the visual system of this animal and we can put together what it looks at in that visual background to decide on its camouflage pattern. How does it do it in under a second? It's got to be a neurobiological shortcut, and that's what we're after. Well, I can tell you right now, after 12 years of experiments every week and a lot of published experimental papers, this bottom animal looking at the checkerboard is looking at the white checker, not the black checker. And when the white checker is about the size of the white square that the animal can put physiologically on its back, it uses that as the simple cue to turn on disruptive coloration. So we can illustrate that a little bit. Underwater on the left, you see a cuttlefish that is all bright and conspicuous. It decides to go into a uniform pattern and disappear. So before and after is pretty different. But now we do a real test in the laboratory. There's a cuttlefish down here at the bottom on a uniform camouflage pattern on a uniform background. We rudely pull the rug out from under it, and it's got a different substrate. The animal checking out the substrate as it swims around and settles right there and puts on a mottled camouflage pattern. And next, what happens, if you look in the top right, you'll see right about now, our special talented three-handed technician rolls in these white rocks from heaven. The animal takes a look at them, moves, and turns on a disruptive pattern because the white rocks are about the size of the white square in the middle of their back. So a simple visual cue, it can overcome all of the other visual information that says stay down here in a mottled pattern, which would have been good, but it switches to disruptive. So there's a priority. So this is not a reflex. Camouflage in this animal is not a reflex. It's a very refined decision-making process. That's the point I'm trying to get across here. So if we look at the skin now, you see the chromatophores through a microscope opening and closing. And so there's direct neurons coming from the brain to operate the muscles that radiate around from each chromatophore. And therefore you can do that in about 100 milliseconds. And here's a diagram from a NOVA show, and you've got a pigmented chromatophore and the muscles radiating out, they're relaxed. The neurons tell the muscle to contract, and it pulls this out into a disc of color, which is now visible. And you have yellows and reds and browns down here, and you have control of all of those layers of color pigmented chromatophores. And then on the next layer down in the skin, you have accentuated here, iridescent green and blue and pink iridophores with a white base layer. This is all structural coloration. So this magnificent, beautiful skin produces the artist's palette with pigments and reflectors. Now, here's a chromatophore. This is an animation done based on real video. And you see, we point out that the skin is always moving. And so the muscles that control it and the pigment sac itself have to accommodate for that movement to give color coherency. And this is why there's a grid underneath and you can see what steady would be. But the animals have figured out how to do that. And engineers are going to have a much tougher time trying to emulate this than they are trying to do this. Speaking of engineers and material scientists, we work with them all the time in all of our grant projects. And during a DARPA project, one of the groups with David Clark, Evelyn Hu at Harvard Engineering have submitted a patent to create display technologies that operate like the squid chromatophore, where you have a punctate color sac and it gets pulled out horizontally at about 100x the change in area. There's no display technology that works like that. So we've also been invited to write articles in places like the information display community. Here's one we did a few years back, quite a few years back, with our cuttlefish on the cover and posing this crazy question, is this the future of displays? Well, it could be in the future. Now, there are pigments in the skin, but now we're going to look at reflectors. Notice here, when I play this again, that the iridescence is gone. It shows up under neural stimulation. And notice the color shift. We'll notice in the next one, it shifts from red all the way across the color spectrum to short wavelength green. And this is due to an extraordinary protein called reflectin. And so you can take this reflectin and you can make other kinds of goodies and biomolecules that are used for optical physics. And some of your alumni from Tufts or current faculty at Tufts have done some of this work. David Kaplan and Fio Omenetto are part of the crew who've done this. They're all part of the biomedical engineering group at Tufts. But you can look at some of these titles looking at that reflectin protein and how it creates optical materials. There are other groups who are looking at some of these tissues, fibers and adhesives for active camouflage, including the cephalopod. So there is an outcome for some of the basic science research we're doing on how the individual chromatophores and iridophores are actually dynamically controlled. So I put an image of an iPhone up here because we all have some kind of smartphone, and I ask you, where does the color and pattern come from? You got a computer, you've got a battery, the battery runs out every day, you drop this thing, computer costs you another grand. And the whole idea is that this machine is creating all the light and pattern. And my point is that our animals can produce all of these colors and patterns, and they're not creating any new pattern or light. They're using available light and manipulating light with pigments and reflectors to get the same result. So the secret here is not just pigments alone, not just reflectors alone, but the combination of the two is very powerful to really fool around with light. So maybe this is a different route towards synthetic materials. So I'm going to show you here, right here is one of those leucophores, that's structural coloration. I take my stimulated electrode and move it on top or to the side or back on top. And the chromatophores on top, the pigments will either enhance or obscure the underlying white reflection. So this is one way to create a synthetic material with two simple processes. But let's take a look at that white, because this turns out to be the whitest white known in the animal kingdom. And we find it in this cuttlefish up to the side here. We look a little more carefully to these white dots in their fin, and we look at it really carefully and we measure with a spectrometer. And when I said the whitest white, here's the full visible spectrum. White is all colors. And so it's producing and reflecting all colors coherently, all 400 colors in the visible range at a high level. The physicist who looked at this said we were exaggerating and that we couldn't do that, but we actually showed them the data, and maybe some of them believe us, maybe some of them still don't, who knows. But here is some electron microscopy where we've isolated one single cell with 12,000 spheres in it that produce this white. And there we've modeled it, and we've emulated it and modeled it, and actually looked very carefully. This is me scattering to give you such an incredible passive arrangement to get pure white. Well, maybe you could produce a better Kindle reading device, and we actually work a little bit with the Ink Corporation right there in Cambridge as another way to use available light more efficiently. But this could also act as a makeup base, because just like in the cephalopods, it's a base upon which the pigment lies on top to enhance it with contrast. But it turns out that this also manipulates ultraviolet light, although we don't know that in precision. But if it did, now you have a translucent protein that will block UV, but we don't know how much of the UV. It's only an idea right now. We haven't measured it. So to move on, this skin, the pigments plus the reflectors give you a remarkable diversity of appearances. And so I was invited to Estee Lauder, the cosmetics company, to give a TEDx presentation. And my point to them was that the future of cosmetics is to have changeable cosmetics that the individual can handle a little bit if you use pigments and reflectors in a sophisticated way, the way the animals are doing it. And here's a high resolution image of cuttlefish skin, and you see the very subtle blushing going on here. And don't you know that women in particular would really like to have this capability. This is not out of the realm of possibility now because we know enough about the biology and the structure and the control of these pigment reflectors to start translating some of that information. The final part of that is the skin texture. You've already seen this video, but here's another one of the giant Australian cuttlefish, smooth skin, periscope up, you got those big bumps. We look at the same animal a little later on, and you see close up that big bump. That's called a papilla, and it's a muscular hydrostat, kind of like a human tongue. It's all soft muscle. And so this is the only animal group on the planet that can change the 3 dimensions of its skin texture. And don't you know, the Navy would like to be able to put bumps on their vessels and their submarines to camouflage them. But then when you have to go fast, you want to put them down flat so there's no hydrodynamic drag. Well, that's how the cephalopods use it for those 2 things. Well, I just want to show you before I show the translation. Here's a giant Australian cuttlefish. It's a big animal. It's, could be 10 kilos. And it's bright conspicuous pattern with smooth skin. And 1 second apart, I shoot it. 1, 2, 3, 4, 5. And you see the animal has morphed into sort of an algae look-alike. Now it comes out of it and you see it goes back to its smooth skin. So it's got great control of the bumps as well. Our colleagues at Cornell, led by Rod Shepard, took our ideas and our publications on how those papillae work, and they made the first synthetic papillae, or let's say 3D texture morphing materials. Down here in the picture is some octopus papillae that we had done. And so we work with them to help them. This was a system that's got polymers, stretchable polymers that are driven by pneumatics. And this was published in Science a few years ago. So, a real bit of translation in a first step, because we don't have materials in society or industry that can do this under full control. Okay, so just to summarize on the complexions, masking harsh edges would be important. I get invited to international dermatology meetings pretty often to talk about the skin because they're always interested in different skin. It's these gradations of color and contrast with pigments and reflectors. And I really tell you, there is a gold mine of biodiversity out there in the natural world with fishes and invertebrates and vertebrates, and certainly our cephalopod invertebrates that use color and pattern with pigments and reflectors. And the vast majority of it is undescribed. So different way to look at the planet. Cool thing is they're manipulating available light. They're not creating light. So there might be some energy savings. Again, our animals have the only shape-shifting skin known to science. In terms of medical applications, vitiligo, there are a lot of them. So just a brief thing about art and science. Here's the same cuttlefish left and right, camouflage or conspicuous. I want to point out that what they're really manipulating in their skin are these factors, which we know, light, shape, contrast. But I want to point out that artists and photographers, architects, landscape artists, makeup artists, marketing and advertising folks are all manipulating exactly the same features. And so if you can look at those and control them one at a time, you get a lot of possibility. Well, the art community is the community that does this. So I teach occasionally at RISD and at Brown with joint courses, and I have RISD students in my lab often. And so we have a real collaboration going with the art community. I currently have an active grant with the Office of Naval Research, and I have 2 RISD professors as part of that grant. Okay, so last thing, almost colorblind camouflage. Look at this camouflage octopus moving along, and it gets to this red kelp, and it goes red, and it goes to skin texture similar to the kelp. Well, the whole deal here is that this octopus and all cephalopods are colorblind. So how do they match the color of the background? And the answer basically is that we don't know, but it turns out that there are opsin molecules in the retina are also distributed in the skin. We discovered that with colleagues. And here's the bottom line. John Rogers, a really accomplished person at now at Northwestern, and his crew looked at our studies of the opsin molecules in the skin. We couldn't prove anything biologically, but he said, Roger, this is a rare chance, or we can actually emulate something you think the animal's doing, but we can put it into our technology. So down at the bottom right here, if you can see my cursor, you see U of I, University of Illinois. That's where John was when he did this work. And this is a piece of flexible electronic material that he plops down on a black rod, and on that black rod underneath is printed UOI. And this piece of flexible material sensed what was underneath it and produced it up top in those little squares with no computer and no power source attached to it. So this was a first, and it was published in Proceedings of the National Academy of Sciences. So here we have a bio-inspired idea really coming to a new capability. So just to wind up the talk, these are exciting times for biotechnology. Probably don't have to tell this audience that. Nature has provided a lot of frontiers that we really haven't even touched very well yet. This is one of them. The oceans are an untapped gold mine still for biodiversity in many ways. Sensory ecology is kind of what I do. I'm interested in how animals operate in their ecological context, limited by or constrained by whatever their sensory apparatus happen to be. And we're never going to understand ecology and conservation until we realize what the world is like to the animals involved. We tend to anthropomorphize way too much. Field work is a key inspiration for me. I've got all my best ideas when I'm immersed in the sensory world of those animals. I do lab experiments only to, let's say, corroborate the things that I've learned underwater. And applying new technology is always really important. One last little saying here at the MBL by Albert St. Georgi: discovery is to see what everyone has seen and think what nobody has thought. It's a nice little fresh reminder that is on a large sign in the building I work in. So with that, I'll conclude with this saying from Agassiz in the MBL library. It's been there since 1888 or 1890. Study nature, not books. Well, at least that's my excuse to go diving. So thanks very much for your attention.
Hi everyone, my name is Amanda and I am the liaison for the Cape Cod Tufts Club. Thank you so much to all of you who have joined to us today. Roger, thank you so much for your fascinating talk. So I'm going to be moderating the Q&A. I originally had plans to go downstairs, but it's lunchtime for my toddler, so upstairs I am. I hope nobody minds. So let's go ahead and jump right into it. Friendly reminder, if you guys do have any questions that you'd like to ask during the Q&A session, please feel free to do so using the Q&A function at the bottom of your screen. So our first question, Roger, is going to come from one of our Cape Cod Tufts Club board members, and I'm reading it off of my phone. He submitted it in advance. Are there any octopus in New England waters, especially in Cape Cod waters?
No. And why is that? And I still don't know because I've been looking. There are octopuses up and down the west coast of North America and much of the East Coast, all the way up to about the New Jersey area and the Carolinas. And then again, way up, you know, in the Arctic, but nothing along here. And no one can explain it. Sorry. That's why I go elsewhere to do my diving on octopus.
Okay, fantastic. All right, here's another one. Does the unique RNA editing you mentioned apply to both coding and non-coding RNA?
I'm not sure because I'm not an RNA specialist. So I would refer that question to Josh Rosenthal at the MBL. He's published a lot of papers on this. So I'm not going to try and wing it with a half-baked answer.
Sure. And can you just define genome for us and how that relates to the information that you were talking about in your presentation?
Well, the genome will give you a layout of every gene that that organism possesses and gives you detailed DNA information. The way we're going to use it, the way we are using it right now, is that I talked a little bit about the reflectin proteins that produce the whiteness and the iridescence, and that's what the Tufts folks were working on. We've all been working with 3 reflectin proteins and all of those reflectors and all the work we've done. And the genome information now has informed us of how much in the dark we've been because there are 18 reflectin proteins in the genome. And we've done enough in situ hybridization to find that there are bits of different reflectins throughout the skin, which we were totally unaware of. So this is going to transform our study of iridescence and whiteness and structural coloration, because now we know there are another 14 reflectin proteins out there in different parts of the skin. And now we've learned roughly where they are. So now it's time to start sorting them out and figuring out how they work and how you can make some new bio-optics toys with them later on.
Great. And so here is another one. I think, Roger, you may have already answered this, but what types of octopi do we have near Cape Cod? And just in general, what type of waters do these animals tend to prefer?
Well, the octopuses are found all around the planet, even thousands of feet deep. There are some deep sea octopuses. There are many tropical coral reef, kelp forest, sand plain, tropical temperate octopuses all around the world. They just don't happen to be here, which is a total oddity. There are about 700 cephalopods have been described. Among those, about 250 are octopuses, but everyone figures there are at least another 100 octopuses yet undescribed. So there are a lot of octopuses crawling around in different habitats on the planet. So it's a very successful group and sometimes very plentiful.
How long in the history of Earth have these critters been doing this?
Well, the cephalopods in their former form, were here in the Cambrian. They literally dominated the oceans all the way back as far as you can go. There were a lot of these things were big shelled swimming things, kind of like modern day nautilus. I think everyone knows what nautilus is. But these were nautiluses that were 6 and 10 feet in diameter and weighed 5 tons. They were like big swimming snails. So the cephalopods have been around, pretty much forever in terms of organisms on the planet. It's the modern cephalopods called the coleoid cephalopods, and they came on the scene. Oh, I guess I should know this number, but I don't. but they're relatively new in the geological record, and so they're the new ones on the scene. And what they did, like they gave they made that external shell a vestigial organ, and they took on a big brain and big sensory systems and this incredible skin and their locomotion. And so they've gone a totally different route from the other mollusks, and a pretty much different route even from most of the, sort of, let's say, ancestral or original cephalopods. So they are really morphing in a very different way, evolving in a different, really different direction in many ways. I mean, these are some of the weirdest animals around. I fully admit it, but I actually like the weirdness.
And I actually would like to ask my own question. Can you tell us a little bit about how you became involved in this world, and what have been some of your favorite places to go diving?
Well, I was a junior in college and went to visit my brother who was in the Army at the time in Panama. And like all brothers, he wanted to get rid of me one afternoon. So he took me over to the coral reef and gave me a mask and snorkel and said, come and get you in a few hours. Don't get sunburned. So I just went out swimming around. It was a very shallow coral reef. It wasn't more than 1 or 2 meters deep, about a meter deep. And I went over this depression in the coral, just swimming along, and something blew water on my belly. And I just about came out of the water. It scared me. And I circled around and just kind of looked down in this depression with corals, and I didn't really see anything unusual. Kept staring at it, and finally saw this little movement. Turns out to be the funnel and the ventilation of the octopus. And I went, wow, this 1 pound octopus scared the daylights out of this 190-pound guy. And then it came out slowly, moved a little bit, and disappeared in front of my eyes. The camo was magnificent. It was only, this far away. I watched it for a full 20 minutes and I was hooked for life. I just couldn't believe that an animal could do that. It was just cool. So I went back, took an invertebrate biology course and did a study on octopus. And I just been stuck on that idea of how they do this camouflage ever since. So camouflage is still one of my main areas of inquiry because it's so complicated and so sophisticated that I've had a lot of fun with it, and I'm still having fun with it. So lucky for me.
All right. So what is the impact of CRISPR of understanding about RNA editing?
Well, I think the CRISPR thing, I don't relate it necessarily to RNA editing, but the CRISPR is going to allow us, in fact, we've already, colleagues at the MBL have already done this. This is Karen Crawford and Josh Rosenthal and Carrie Albertin. And what they've done is they've knocked out the initial enzyme that gives you the pigment in the chromatophore. And so they've done that at the 2-cell stage in the embryo, and then they let the embryo grow, go through the developmental stage and let it hatch out as a tiny squid. And then you see this beautiful illustration of half the squid has chromatophores, half the squid does not have chromatophores. So you've knocked out specifically the pigment sac that creates the pigment only. The chromatophore is not a single cell type, it's 6 cell types. It's actually a complex organ. And so the other 5 cells are all there, and it's only missing the pigment. So now that gives you a way to study the dynamics of the chromatophore with the nerves and the muscles and the sheath cells and all the rest. Without the pigment being present, you can see more and you can see the effect of it. So there's an example of knocking out one gene that gives you the startling effect in the appearance of the animal. So the next thing to do would be to do some knock-ins, or you can knock out or you can knock in. And CRISPR is a technique that allows you to sort of play that game and probe a little bit and find out where things are and where they aren't. So the next step we'll do is we'll try to knock out one of the reflectin genes that we know produces bright red and blue iridescence, and see what the effect is and what the remaining cell can do, and at least find out where it's localized. It'll tell us a lot about optics and the spectrometry. So, and the protein chemistry that's all creating that light interaction. So that's with the CRISPR technique. And, I mentioned in my talk, technology innovations, like we're using hyperspectral imaging now to look at color matching. That's really high-tech cameras. CRISPR is a high-tech and new technique that allows you to play around with genes in a very specific way. So it's just another tool in the arsenal to dig down and learn something about how something actually functions.
Awesome. Here's another one. What studies have been done on the octopus eyesight, and how does that contribute to its adaptive ability? Any ophthalmological lessons learned from them?
Well, I don't know about the ophthalmological lessons, but I will tell you that the eye has been studied in reasonable detail. The octopus and its relatives have very high visual acuity. Um, when you do a cross section in a light microscope of an octopus eye and do it right next to a human eye, they are so similar, it's scary in their gross organization. But their fine organization is tremendously different. The octopus eye only has one photoreceptor cell type. In other words, they don't have cones and they don't have rods like vertebrates do. Without the cones, they can't see color because they've only got one cell type and they only have one opsin tuned to about, what is it, about right around 400 nanometers. So the idea is that they see the animal, they see the world in a monochromatic way, but strangely enough, they can see polarized light because their photoreceptors are orthogonally arranged. So they can pick up the polarization aspect of light. Humans can't do that. So what an octopus sees and what you and I see is tremendously different. We both have good visual acuity, but they don't see color and we do. We don't see polarization and they do. And what that looks like is baffling. But we have to remember not to make too many analogies between what we see in the background and what they see, because it's got to be different. They can handle the information well. So I'm sorry, I can't give you a connection to ophthalmology because I can't think of any at the moment, but I at least was able to tell you a little bit about the eye. Oh, by the way, they have superb night vision too. You go at night and it's just like daylight. We've done experiments with cuttlefish in nature and also in the lab down to 0.003 lux. That's starlight. We are totally blind in starlight only. And the cuttlefish, you put different bottoms underneath and they change patterns just like it's high noon. So their night vision is really superb in addition to having good acuity and good polarization sensitivity.
Fascinating. So these questions are really rolling in so far. We have 27 questions that people have asked. So this is awesome. Keep them coming. So here's another one. Why is the genome of the octopus so much larger than humans? Is it all related to the RNA editing or is it something else?
Well, there's undoubtedly a lot of junk in there, like there are in most genomes. But the answer is no one really knows. Their Hox genes are organized in a very different way. I know that. Carrie Albertin published that in her Nature paper a few years ago. But I'm not a genome expert. I'm a genome dummy. I can't really answer such a question. But I think the genome is so recent that the questions are just being addressed. No one did a full genome until 4 years ago. And now we have the squid genome. And so I know some of the folks in my lab, I've mentioned their names already, they're studying this all day long. And so I'm going to have to wait for the answer to that one. Maybe that can be next year's Tufts Talk. I'll be happy to volunteer a colleague. But let's go fast. If there are 27 questions, let's roll. I'll make shorter answers.
So what is an example of use for camouflage skin?
Well, you mean human? Well, human applications are obvious. A hunter or a SWAT team member or a soldier would love to have better camouflage. I personally have a lot of military background and I would like our troops to have better camouflage. But I think that's one thing. The other is just learning how skin works is another big deal, and how it's controlled. And the nature of the camouflage patterns is something, and how they use their camouflage patterns is really more dynamic than any other animal on the planet. So we're adding to camouflage theory in a big way, and we deal a lot with that. Next.
Do octopi form communities?
No, except one species might in Australia. So octopuses are asocial, solitary animals. We know that for dozens of species, but there is one species that tends to aggregate where their habitat is there, mainly collections of shells from fishermen dumping them in one place. And they seem to tolerate each other, but that doesn't mean it's sociality. That site is being studied in Australia, but it would be the one-off occasion. Basically, the answer is no, there might be an exception or two. I hope there is, but we don't know yet.
All right, so here I'm going to consolidate two questions that came in related to a similar matter. How do you feel about eating octopus, and what is the best octopus to eat?
Yum. The octopuses are too good. I do eat them very rarely. Part of the reason I only eat them in certain parts of the world where I know they're still plentiful and the fisheries are not in jeopardy. And that's true in most places. So that's the good news. Same with squid. Next time you have calamari, you can think of this beautiful skin, but it won't look very beautiful on your dinner plate. But it does taste good. So no, I don't have a problem eating these animals, at least the ones that I know the fisheries are in good shape. I did a lot of fisheries research, so I keep abreast of that. So I'm not overly concerned about that personally. I know some people are. Some people will be offended by my answer, but I'm just being truthful.
Here's another one. How is climate change and commercial harvesting impacting their habitat/prospects?
Well, commercial fishing is probably the biggest problem, not climate change per se, but commercial fishing with bottom trawls really alters the habitat. And for bottom dwelling animals like cuttlefish and octopus, and even squid spawning grounds here on the Cape, that is a big problem. So there is an effect of the fisheries on habitat use of these animals. So there's no question about that. Climate change, the real change investigators are concerned about is that you get the changes in carbon dioxide and acidity and so forth. And we know that if the pH of seawater continues to go down, the cephalopods are quite sensitive to that because in their respiratory system, they don't have hemoglobin to transport oxygen on molecules. They have hemocyanin, which is very pH sensitive, unlike fishes, which can take lower pH. Our animals cannot do that very well. So there's a lot of experimentation going on that at the moment. That's the one thing I think some folks are worried about.
So here's another one related to your background. I know you already shared info on this, but here's a second component to the question. What were you studying before you got into this?
Well, I was in college and I started out studying hotel administration. Who knows why? I mean, I was more interested in sports in college than academics, at least at the beginning. But then I just got interested in biology. I've always been an outdoor kid, and I just gravitated towards it and started taking biology. So from my sophomore to my senior years, I just took biology courses. But then it was Vietnam era and I was in the Army and then I traveled and I went back to graduate school more than 3 years later. So I was not totally committed. I thought I was going to be an underwater photographer as a profession because I got into that and after the Army, diving around the world, but then I sort of glued them together and the biology won. So here I am, but I'm still doing my underwater cinematography and videography. So I sort of combined those two passions that arose. Love of nature and being outside in the water and imaging.
What is the largest size of the octopus and where are they located? And similarly, what's the smallest?
The largest one is the GPO, the giant Pacific octopus. That's Octopus dolphini. It's in the area of Oregon and Seattle in the US up through Alaska and over into Japan. So it's a North Pacific cold water species. A little controversy about how big it gets, but they can get up to 100 pounds pretty regularly. You can see those in public aquaria, including the New England Aquarium. The smallest octopus is a tiny little pygmy octopus called Octopus jubini. It's, I don't know, it's about the size of one of my tip of my fingers and a few small arms. So it's a really tiny little dude for sure.
Here on Cape Cod in the spring, there is a 1 to 2 week of fishing for these animals, I believe, ? What is that all about? Is it related to breeding? Also, what is their ink composed of and how unique is it?
The ink is melanin. It's one of the purest sources of melanin. And also it's delicious in food because the Mediterranean folks, calamari insutinta is calamari baked in the ink. In terms of the squids, there is a squid fishery in the Northeast. It's a substantial fishery, about $30 million a year right here in northern New England. And you do see the boats out here getting squids, especially in May, but also throughout the summer. And the squids have come from offshore in May when the coastal waters get warm enough, and they come in large numbers all the way up into the Vineyard Sound, Nantucket Sound, and they lay eggs. These are all mature squids. They've overwintered in the deep canyons off the coastline here. These canyons, they spend their whole winter at about 900 to 1,000 feet down in these canyons on the edge of the continental shelf. And that water is about a stable 10 degrees. Sounds cold to us. That's like heaven to a squid. That's the perfect temperature for a squid. And they'll come up on the shelf to lay eggs. And then they'll lay eggs. Those eggs will hatch out. And these squids, they only live 9 to 12 months. That's their whole life cycle. So they grow very fast. So the squids you see here around Cape Cod by September and October and November are young of the year, and some of them are pretty big. So they can really grow super fast. And now, as the water's just gotten cold, the squids literally right now are moseying their way off of the Cape because the local water temperatures are coming down, and they're heading off to those deep canyons. Humans go to Florida for the winter, and squids go to the bottom of canyons.
Have you ever been entangled with an octopus?
A little bit. I've had one on my arm. I've had another one crawl up my arm, and it was hard to get it off. You got to dislodge the suckers because you have to worry about it because an octopus can bite you, and they do have a venom, a neurotoxin. Most octopuses will just give you a sore arm and a little bleedy bite. The blue-ringed octopus, which occurs in Indonesia and Australia, which is a little sucker with iridescent blue rings, and its bite will kill a human and has killed some humans. It's almost impossible to get them to bite you. The humans who've been killed have done ridiculous things, like a drunken soldier that was the case that everyone knows about, kept taking this octopus and actually stuck it under his armpit, showing off to a crowd of kids, I guess. And the thing finally bit him after 5 minutes and it killed him. So the neurotoxin for one species is really, really will paralyze the whole human nervous system. But most octopuses are not that dangerous, and I've, kind of mingled with them a little bit. I almost got grabbed by a Humboldt squid once, which was almost as big as me in Baja, California. That did scare me plenty. It only came up temporarily and got it away. So that's an animal I'm definitely afraid of.
Where's your favorite place to go diving?
I've got about 7 favorites. They're all over the globe. The Plow Islands in Western Micronesia are right up there. I've just completed 2 studies in the last 3 years in parts of Indonesia, especially Eastern Indonesia, the Raja Ampat area, which is a couple thousand square kilometers of pristine coral reef and uninhabited islands. These are places that have not been influenced by human dredging and activities. The corals are in good shape. Another favorite dive spot, I have several in the Caribbean where I go where I can find my animals. That's basically where my favorite sites are. Coral reefs, kelp forests off California and off New Zealand, kelp forest off Northwest Spain, which is cold water diving too. So some of the places I'm really going where I can find either the cuttlefish or the octopus or the squid that I'm funded to study, I go to where they are. And most of these places are not very convenient. Some of them are beautiful and I like those. And most of my diving sites are cold water sites. So I'm usually in a dry suit. I guess I'm not smart enough to do more coral reef work, although I do some of that as well.
What are your thoughts on the documentary My Octopus Teacher?
I thought it was excellent insofar as it really brought attention to the beautiful ocean, how sophisticated an octopus can be, how beautiful it can be. and I thought it was a neat story. The story was about a human and sentimentality. That part of it was all really attractive and good. The interpretations of the gentleman about the octopus behaviors, a lot of problems there. They just weren't science-based. And so I've been asked to comment on that one. I like the film a lot, and I really admire that guy's diving abilities. And he did something that I do a lot of, and my colleagues, which is go down and spend a lot of time there and let one animal habituate to your presence. That's how we do all of our field studies. So what he's doing is nothing new. It's just that I've never known anyone to do it by holding their breath, which is a little counterproductive for science because you wouldn't be able to spend enough time to get anything. But I do it with scuba and other toys. But admirable. I think it's going to bring a lot of attention to the ocean, how beautiful it is, and how cool these animals are. The one story you shouldn't believe, though, is the octopus tactically figuring out that if it leaped on the back of the shark, it wouldn't get it eaten. Sorry, that one was fabricated. But the rest of them were pretty darn good. I really like the film, though.
Light only penetrates a couple of 100 meters. Do any deep ocean animals below this depth also have these amazing features?
Yes, but in a different manner. So below where light goes, you have bioluminescence dominates those parts of the oceans where animals communicate visually. And there are many cephalopods, squids in particular, also some deep sea octopods that have all kinds of amazing arrangements of photophores that create bioluminescence. And some of those are used for camouflage, but it's not been well documented. I showed you a uniform motile and disruptive pattern, which are 3 pattern types in light that cuttlefish and octopuses use. And I see something fairly equivalent to the patterns of the photophores on some deep sea squids. But it's hard to study those. But they are definitely using the photophores and communicating either what they are for sexual attraction or what they may not be, sort of a masquerade, we call it, or mimicry of something else. And so there's a lot of stuff going on with light, but it's really weird because it's bioluminescence created out of the skin.
Could you list a few more of the more interesting implications for humans in the future?
For cephalopods. Well, I think that, just looking at what some of my colleagues are doing, one of the implications we already talked about was, devising better camouflage, but I think optical devices in particular are where most of the applications will come from, especially making fabrics that change color and pattern based on how these animals arrange their pigments and reflectors and how they control them. I think that's probably the biggest outlet. But those reflectin proteins that I keep referring to that create the structural coloration and the iridescence, there are some folks who have taken those and they've made electrical devices. You can get because they're kind of a protein pump, the way the protein is set up, it's really or a proton pump from the protein. You can actually generate some electrical activity, and some of those have actually been published. So you just never know what people are going to come up with when they find something. The folks at Tufts took the reflectin protein and embedded the protein into silk fibers, and they made optical devices through silk that could change color on the basis of the cephalopod protein. Now that is some far out thinking right there. So, it's human ingenuity. Once you give them something that does something with light, people are going to figure out how to play with it. So I think those are some of the, I think those are the main avenues that I've seen people working on.
So we are just coming right up on the 1 o'clock hour. Dr. Hanlon, if it's okay with you, can we ask the last few questions that have come in?
I'm not in a hurry. I'm fine.
Great. Okay. Is the octopus endangered, and what is their most serious enemy?
They're not endangered. The biggest octopus fishery in the world is off the Sahara Bank of West Africa. It's a big continental shelf where they're trawled, and it's a big fishery where they get octopus and cuttlefish. The numbers are down, but they're not anything on the red list. There may be one other fishery, squid fishery, that seems to have some of the numbers going down, but thank goodness with their short life cycle and the very high fecundity, the squids in particular reproduce in large numbers very quickly. And I think this, and their mobility is amazing. I mean, it's just pretty hard to catch squids, certain types of squids with traditional nets and so forth. Octopus fisheries in particular, most of the octopus fisheries are very small fisheries. There are artisanal fisheries where communities go out and go down and grab them by hand or in pots. And those are small fisheries that only feed small communities, and they're managed well enough, and the octopuses are they're available enough to reoccupy. The octopuses have most of them have a larval stage coming out of the egg, so they disperse a lot. And so that means that they disperse a lot, and there are a lot of animals that are settling on different places. So remove some octopuses, others settle, and there seems to be, I wouldn't say an equilibrium, but there's no area that I know of where they're really being fished out. The one cephalopod that is definitely being fished out is nautilus in areas of the Philippines. Nautilus is a deep water animal. They put traps down there, and some of the nautilus are on the red list. Don't anyone go out and buy any nautilus shells cut in half to decorate your home. That is the reason that the fishermen go after them and sell them around the world. And they're finally on the red list because they have been fished with impunity. And so that's the only one of the cephalopods that is really in some trouble in most of that area of the Philippines.
What has been the cosmetics industry response to your research?
A lot of interest, but no action yet. A lot of discussions. And so they say, yeah, well, that makes sense. We could talk about that. But I think their R&D sections have not yet been ready to go in that direction. We were getting close for a while, but I haven't had any discussions with them recently. So, some very influential people in that community have talked to me. They realize that there's good enough science behind it to do something with it. But they're not funding agencies. Yes, they do fund some research, but it's a little different. So nothing too much has happened in that area yet, but I think it could come around in the next decade. We'll see.
Are octopus creatures more concerned with their own safety / needs and staying out of sight?
Yeah, but what animal isn't? I'm not so sure about that question. Yes, I mean, an octopus is a solitary animal. I mean, a day in the life of an octopus is pretty tough. I mean, they're hiding in a hole most of the time because they are so vulnerable. I mean, I sometimes describe an octopus and define it as a yummy hunk of protein swimming in the ocean that everything's trying to eat. They're right in the middle of the food chain, and every toothy predator out there and diving marine mammal and diving birds are trying to get octopuses. So they have to be really careful when they're out, which is one reason they have such great camouflage. And so finding food is a big deal. So they're going out and they're parachuting over rocks and getting everything underneath. They're getting the occasional crab, and they rarely eat fish. So an octopus life is really tough. They're hiding most of the time. They're daring to go out to the grocery store and not get killed. I mean, it's equivalent to that. And that's it. So they don't have much time for play. These are smart animals insofar as they have short and long-term memory. Their spatial memory is probably among the best in the animal kingdom. They'll go all around a coral reef for 2 hours and go 80 or 100 meters in the most complicated 3D environments on the planet, and they'll find their way back to a little hole in the ground. And we divers are always confused and lost. We follow the animal and it plops down in a hole, and we realize that was the same hole we started at 2 hours ago. We've actually published that and shown how complicated it is. So that takes a lot of brain power. So they're really good at some things, and then other things, they don't do because it's not part of their lifestyle.
Could you describe the importance of kelp and the current condition of kelp forests?
Yeah, I'm not thoroughly knowledgeable about it, but the kelp forests come and go with environmental changes. Right now, the kelp in California really went through a really bad period. And New Zealand went through that for a while too. Back in the 1970s, there were big problems with kelp forests because the sea urchins were eating the base of the kelp, and the kelp were then floating away and dying. And so these cycles come along where one animal, in that case, the sea urchins, were implementing it. Other times, temperature can have a big effect. You know, kelp beds in California during El Nino versus La Nina are very different. They're temperature sensitive in terms of how abundant they are and how fast they grow. But I don't keep abreast of all that literature, so I can't say much more than that, except the kelp situation in California in the last year is really different. And so maybe someone can follow that up on the web with the California situation.
What is the difference between a cuttlefish and a squid?
It's a big difference. The cuttlefish and the squid, unlike the octopus, they have 10 appendages. They have 8 arms, but they have 2 tentacles, which are hidden all the time. And they only use them to flash out at high speed to grab a prey organism. A squid is a pelagic animal. It's always swimming out in the water column. A cuttlefish is not out there swimming in the water column. They're down near the bottom. They're on the bottom or swim close to the bottom. So their habitats are very different. A squid is negatively buoyant. Day and night, it has to pump to stay up. So it's expending energy like mad. A cuttlefish doesn't do that at all. A cuttlefish has a cuttle bone, and it can pump gas into the bone or out of the bone. It's not a true bone. It's called a cuttle bone, but it's calcium carbonate. And it pumps gas in or out to give it neutral buoyancy all the time, no matter whether it's deep or shallow. That's really cool. So that's a major difference between a cuttlefish and a squid.
All right, 2 more. Here we go. The near instantaneity of these color changes is amazing. Any idea how this communication across the cells occur?
Yes, we have a good idea because, and we've done a lot of work, we and our colleagues, the individual chromatophore organs in the skin of a squid, if you look at the neurons that are connected to that chromatophore and you follow them backwards, you stain them with, different things that stain neurons. Hydrogen peroxide is one of these. You can follow that neuron all the way back, and we and others have done that study. And the cell body of those neurons is all the way back in the brain, which means that you've got the fastest system possible in a biological system. The cell body is in the brain and sends the signal out through one neuron all the way to the end organ chromatophore to turn it on. It's like a light switch in your house. You can't make anything faster in biology. Other animals change color by putting a hormone in the blood system, and the blood system has to distribute it through the blood system. And all of that takes time. It takes minutes, days, hours. If you want something to go fast, you get direct control or direct connection by the nerves. There's not even a nerve-to-nerve junction with a synapse in between. They don't even have that in the cephalopods. It's just one long nerve that sends the signal down as fast as you can possibly get it there. That's the system. It's really unusual.
That's really interesting.
It's fast.
All right,
Yeah.
so here's our last question. Why do you think octopuses have multiple hearts? Unsure of what their circulatory systems are like, but must take a lot of blood/nutrients to support all their neurons and many legs.
Yes, well, the anatomy of these animals is unusual. They have a central heart, like many animals do, like us, but they have at the base of each gill, they have 2 big gills, and at the base of each gill, they have another heart, and that heart pumps the water through the gills. So there are kind of 2 things going on. One is the central heart isn't big enough to push blood through the total vascular system. And second of all, it seems as though each gill has enough back pressure that it requires an extra pump right there to push the blood through those gills and then to push it out into the arteries throughout the body. So this is one of the oddities of these animals. I mean, if you play Trivial Pursuits, one of the questions is, how many hearts does an octopus have? So the answer is 3. You can get that one right. But it's quite an inefficient system, but it's an oddity more than anything else. It worked great for the cephalopods. Why other animals don't have accessory hearts at the base of their gills, I don't know the answer to that, but I'm sure it has to do with the physics and the resistance in the vascular system.
Awesome. Well, so that concludes our live audience Q&A session. Dr. Hanlon, thank you so much. And to all of you who submitted questions, thank you to all of you. I have to say, in all my time of doing this, which has been now nearly 8 months, this has to be one of the most engaged audiences that we've had. So kudos to all of you. This has been really, really enlightening, and thank you all for tuning in. And without further ado, I'm going to pass this back to Mike Duffy.
Okay, can you hear me, Amanda, and everybody else? Okay, I don't see myself. Thank you, Dr. Hanlon, for that captivating presentation. And to our attendees, we appreciate your participation and your thoughtful questions. You will receive an email next week with a link to a brief 2-minute survey, which we hope you will complete. And as I mentioned earlier, this event has helped us raise $760 for the Cape Cod Tufts Club's Scholarship Fund. If you are so inclined, we hope you will consider making a gift to this fund. Amanda will post that link now so you can do so online. Finally, I would like to invite you all to join us again on Wednesday, December 16th at 7 p.m. for our virtual holiday concert. So save the date. An email for registration will be sent to you soon. Have a wonderful rest of the day, and please stay safe and be well. Thank you.